We do see cases of deep divergences within species. Here's some examples in North American Lepidoptera, a member of the genus Lepidoptera, a Splusia species, a Hyphanthria species. When every one of these cases is looked at carefully, they turn out to be cryptic species. This is the rice and corn variants of this genus. This individual feeds on blueberry. These feed on grasses. These caterpillars have different head colors. So there are many overlooked species in well-studied groups like Lepidoptera, and when you move into organismic lineages like parasitoids and surely nematoids, we will see an explosion in species count, order of magnitude elevation. So there aren't, I think, deep intraspecific divergences. When you see it, it is due to a failure for the current taxonomic system to recognize biologically, reproductively isolated lineages. So over the course of collecting these data, this is, I think, something that we say has generality in environments across our planet in organismic lineages. Common observation of almost no variation among individuals, or very shallow, deep divergences within species reflecting cases of overlooked taxonomy. Between species variation, cases like this do exist, but they're rare. They're 1 or 2%, and most of these cases involve situations of hybridization, where there's been some sharing of mitochondria. Shallow divergences occasionally, and deep divergence is the rule, 95% or more. There's a potential zone of overlap. You simply can't use a hard threshold and use that to say this is a species, this isn't a species. But in the large majority of cases, the divergences are very informative. If you find a divergence that's several percent, it's a different species. So that's sort of the ground truthing studies that have gone on. What about all the life on the planet that we haven't looked at intensively? Can we use these ground truth studies to develop rule sets that we might use to probe life at large? In particular, can we use barcodes as an additional screen for overlooked species? And I'll argue that we can, but because there is such a close correspondence between, in the ground truth systems, in the birds and fishes, there's such a close correspondence between barcode distinct lineages and those species recognized by the taxonomic enterprise that we can do this. Let me just show you an example. Here's some little species of Lepidoptera from the temperate zone. Sequence divergence. These little orange lines represent 1% sequence divergence. And you can see that every one of the recognized species in these groups here in Ontario is easily recognized. And you can see the deep branches. In fact, if we use a screening threshold of 1% and 1%, every 2%, every one of these things are recognized. Start driving back along this axis and you can see what happens. You start recognizing fewer and fewer species. And once you get back here, if you use a screening threshold of 20%, you don't recognize 2 species. But thresholding, a 2% sequence threshold here, reveals all of the species the taxonomic enterprise has revealed. Let's look at things a bit more generally. And I find these curves very, very interesting. These are the birds of North America now plotted as the number of genetically distinct groups you will find if you look at 2,500 bird specimens and start slicing at various thresholds. And you can see you'll find an enormous number of genetically diverse groups at very low sequence thresholds, fractions of a percent. Now we're driving out to increasingly strong thresholds, just like I showed you with the insects. Note the collapse. This blue curve is the within species variation. The linear curve are the different species. These are the thresholds. This is birds, about 1.5%. There's this inflection point. These are the things that we recognize when we use these hard thresholds. The really cool thing is if species are being born at a uniform rate, we can project that. We can actually see these things that are invisible on a threshold, and we can say how many species of birds there are in North America. Lepidoptera of North America, Oz fish, Lepidoptera of Costa Rica. The curves are incredibly similar. This linear part, that's the birth of species. This exponential decline that's due to the stripping out of variation within species. These two very distinct processes. So I believe these rules are startlingly similar across different groups of life, and they're going to allow us to probe the unknown universes of organisms on this planet and reveal the diversity of nematodes, reveal the diversity of any protists, all those unstudied groups. So, is everything perfect? No. There are groups that show low rates of evolution. The cnidarians, benthic cnidarians. We have to add fast evolving genes to get species level resolution. Can we recover barcodes 100% from every organism we encounter? No. In the groups we worked hard, we can. Not in all groups. There's a need for these detailed analysis of splits. We have to get the biological covariates to understand and verify. We're not describing species on the basis of barcodes. We're saying these are situations that demand investigation. And then finally, I think, we need to register these species in the Linnaean lexicon. We need to fast-track species descriptions. We need a new protocol that simplifies this whole process and move things out of the interim names into a more formal Linnaean system. I think we can do this, and we're finding some appetite for people to join in this force. So I'm thinking about a streamlined mechanism that's really the parallel for species descriptions that barcoding is for probing genetic variation within lineages. So as a summary, on the animal front, about 98% resolution, I would say, of known species. Why does it work? Young species are rare, and selective sweeps are taking out intraspecific variation, building this chasm, a difference between congeneric diversity and within species. And species counts, I think we can go into unknown universes and accurately predict. It's not going to be perfect, but I think we have to get away from the notion of perfection in barcoding. It's not a perfect technology. It's an accurate technology that's delivering, I think, quite strong insights that will be very valuable in many contexts. And I think that was my last slide. Thank you.